Hello students, this is Dr. Kausalya, Professor of Food Science and Nutrition from Avinashalingam Institute for Home Science and Higher Education for Women. In today's lesson, we are going to see the concepts of genomics and proteomics. Earlier, in the lesson on recombinant DNA technology, we have studied about how the transcription occurs, how the recombinant DNA is formed. In today's lesson, we are going to see about the genomics and proteomics. Genomics is an area inside genetics that relate the sequencing and analysis of an organism's genome. It is the entire DNA content that is present within the cell of an organism. Genomics experts attempt to find out the complete DNA sequences and perform genetic mapping to help and identify it with disorders. So what is it that we are going to study in this lesson? This lesson involves the study of intragenomic processes like pleiotropy, heterosis and epistasis with the interactions between the loci and alleles within the genome. The fields of molecular biology and genetics are mainly concerned with the study of the role and function of single genes, a major area in nowadays which is very much important in biomedical research. Genomics does not involve single gene research unless the purpose is to understand a single gene's effects in context of the entire genome. The objectives of this lesson include to introduce the concepts of genomics, to describe the concepts of proteomics, to study the different types of genomics and proteomics, and to see the applications of genomics and proteomics as two different fields. As per the definition from the United States Environmental Protection Agency, genomics concerns a wider line of scientific inquiry and associated techniques than it did initially. Genomics involves the learning of all genes at the DNA, proteome and mRNA level with the cellular and tissue level. Genomics concept was first developed by Sanger, who first sequenced the complete genome of a virus and of a mitochondria. He did mapping and he initiated the practice of sequencing and genome mapping with budding bioinformatics and data storage in the 1970s and 1980s. The knowledge about genes that has so far been gathered has led to the coming out of functional genomics, a field concerned with trying to understand the pattern of gene expression, especially across different environmental conditions. The term genomics was first coined by Tom Roderick in 1986, a genetist at the Jackson Laboratory in Maine during mapping of the human genome. What do you understand from the term functional genomics? It refers to the study of molecular biology that attempts to build the use of huge wealth of data produced by genomic projects to explain gene and protein functions and of course their interactions. Functional genomics focus on the dynamic aspects like gene transcription, translation, protein to protein interactions as different to the static aspects of the genomic information such as DNA sequence or structures. Functional genomics attempts to answer questions about the function of DNA at the level of the genes itself. RNA transcripts and protein products too are involved in these. A key characteristics of functional genomic studies is their genome-wide approach to these questions, generally involving high throughput methods rather than a more traditional gene by gene approach. What do you understand from the term structural genomics? This explains the three-dimensional structure of each protein encoded by a genome. This genome-based approach allows for a high throughput method of structure determination by a combination of experimental and modeling approaches. The principal difference between structural genomics and traditional structural prediction is that structural genomics attempts to determine the structure of each protein encoded by the genome rather than focusing on one particular protein. Structural genomics involves taking a large number of approaches to structure determination as well as experimental methods using genomic sequences or modeling based approaches based on sequence or structural homology to a protein of known structure or based on chemical and physical principles for a protein with no homology to any known structure. As different to traditional knowledge or traditional structural biology, the determination of a protein structure through a structural genomics effort often comes before anything is known as protein function, and this is with relevance to the genomics. This raises new challenges in structural bioinformatics, that is, which determines the protein function from its 3D structure. You see an example of a protein structure determined by Midwest Center for Structural Genomics. Students, now coming to the concept of epigenomics. It is the study of epigenetic modification across the entire genome. An epigenome consists of chemical components and proteins that can attach to DNA and helps in the production of proteins in particular cells, or it may either control the whole mechanism. DNA methylation and histone modification are the most characterized epigenetic modifications. Epigenetic modifications play a vital role in gene expression and regulation, and therefore are involved in several cellular processes like differentiation, development, and tumorigenesis. The study of epigenetics on a universal level has been made possible only recently through the adaptation of genomic high throughput assays. What is metagenomics? It is the study of metagenomes, the genetic material recovered directly from environmental samples. It may also be referred to as environmental genomics, ecogenomics, or community genomics. Through traditional microbiology and microbial genome sequencing, which depend on cultivated clonal cultures, early environmental gene sequencing cloned specific genes, often the 16S RNA gene is involved to produce a profile of diversity in a natural sample. Recent studies use shotgun Sanger sequencing or massively parallel pyrosequencing to get largely unbiased samples of all genes from all the members of the sampled communities. Because of its power to reveal the earlier hidden diversity of microscopic life, metagenomics offers a powerful lens for viewing the microbial world that has the potential to develop understanding of the entire living world. So what are the model systems that are used in this? Let us see about viruses and bacteriophages. Bacteriophages play a key role in bacterial genetics and molecular biology. Previously, they were used to define gene structure and gene regulation. Also, the first genome to be sequenced was a bacteriophage. Still, bacteriophage research did not lead the genomics revolution, which is clearly dominated by bacterial genomics. Coming to the category cyanobacteria. At present, there are 24 cyanobacteria, which a total genome sequence is available. 15 of these cyanobacteria have come from the marine environment. Quite interesting, isn't it? There are 6 Pyrochlorococcus strains, 7 marine Synechococcus strains, Trichodesmium strains, and Crocosferia strains. Some studies have shown how these sequences could be used very successfully to infer important ecological and physiological characteristics of marine cyanobacteria. Yet, there are many more genome projects currently in progress. Amongst those, there are further Prochlorococcus and marine Synechococcus and, of course, Eukaryochloris, Prochloron, and the nitrogen fixing filamentous cyanobacteria. Limbia species, the different types of species, as well as bacteriophages infecting marine cyanobacteria. Thus, the growing body of genome information can also be tapped in a more general way to address global problem by applying a comparative approach. Some new and exciting applications of progress in this field are the identification of genes for regulatory RNAs, insights into evolutionary origin of photosynthesis, or estimation of the contribution of horizontal gene transfer to the genomes that have been analyzed. Nutrigenomics. What is nutrigenomics? We have seen separately this in an earlier lesson, but nevertheless here it bridges both even the epigenetics factor, the genetics factor, and also the proteomics factor. So let us recall what is this. Again, coming to nutrigenomics, it's a branch of nutritional genomics and is the study of the effects of foods and food constituents on gene expression. It is a field of research focusing on identifying and understanding molecular level interaction between nutrients and other dietary bioactives with the genome. The human genome is estimated to encode over 30,000 genes, can you imagine, and to be responsible for generating more than 1 lakh functionally distinct proteins. While traditional nutrition research has dealt with providing nutrients to nourish populations, now there is a shift that focuses on improving health of individuals through diet, while modern nutritional research is aiming at health promotion and disease risk reduction and on performance improvement. Nutrigenetics questions as to how individual genetic disposition manifesting as single nucleotide polymorphism, copy number polymorphism, and epigenetic phenomena affects susceptibility to diet. Nutrigenomics addresses the inverse relationship, that is, how diet influences gene transcription, protein expression, and metabolism. Metabolomics is a diagnostic tool for metabolic classification of individuals. A major methodological challenge and first prerequisite of nutrigenomics is integrating genomics, that is gene analysis, transcriptomics, that is gene expression analysis, proteomics, that is protein expression analysis, and metabolomics, metabolite profiling, to define a healthy phenotype. Hope you are able to bridge all these concepts. Nutrigenomics may offer a new approach for understanding the beneficial effects of dietary compounds on the development of severe polygenic diseases like hypertension, diabetes mellitus, and cardiovascular disease. Nutrigenomics consists of 3 omics disciplines. They are gene, protein, and metabolite, We have seen the definitions earlier as transcriptomics, proteomics and metabolomics as applied to the field of nutrition and health. As one singular factor, they must work for nutritional systems biology that involve the understanding of active interaction and balance between food components and the entire diet with cells, organs and the whole body as a system. Nutrigenomics thus forms the scientific basis for developing nutrition adapted to the specific needs of consumers. Genetic susceptibility may predispose an individual to a diet-induced disease and or render this individual more or less susceptible to dietary interventions. These polymorphisms predispose individuals of different ethnicity to having a high risk of developing hypertension, atherosclerosis and of course metabolic syndrome too. Coming to the concept of proteomics. The word proteomics was termed by Mark Wilkins in 1994. The word genomics is thought to have been formed in reference to the word chromosome. Genomics refers to the analysis of all genes and transcripts in an organism. Proteomic and metabolomic techniques are now enable us to find out more about the basis of the associations through examination of the functional interactions of food with genome at the molecular, cellular and systemic levels. Even though all cells in a multicellular organism have the same set of genes, the set of proteins produced in different tissues is different and it is dependent on the concept of gene expression per se. Thus, the genome is constant, but the proteome varies and is active within an organism. In addition, RNAs can be alternatively spliced. Spliced means it is cut and pasted to create novel combinations and novel proteins. And many such proteins are modified after translation, which is called post-translational modifications. Although the genome provides a blueprint, the final architecture depends on several factors that can change the progression of events that generate the proteome. So proteomics complements genomics and is useful when scientists want to test their hypotheses that were based on genes. It is a term that is used to describe the whole set of proteins as I mentioned earlier, and it can be understood using the knowledge of genomes, since the genes usually code for mRNAs and further the mRNAs encode proteins. The entire set of proteins produced by a cell type is known as a proteome. Genomics led to proteomics via transcriptomics is a logical step and it is a directional step too. Proteomes can be studied using the knowledge of genomes because gene code for mRNAs and the mRNAs encode proteins. Although mRNA analysis is a step in the right direction, not all mRNAs are translated into proteins. Even though all cells of a multicellular organism have the same set of genes, the set of proteins produced in different tissues. In contrast to the genome, the proteome is highly dynamic and constantly changing in response to the environment of a cell or an organism. Comparing such variations in the proteome enables the discovery of key proteins and the identification of modulated pathways involved, for example, in specific nutrition related processes. Protein-protein interactions which complicate the study of proteomes. Although the genome provides a blueprint, the final architecture depends on several factors which changes the progression of events that generate the proteome. Proteomics is evolved as an analog to genomics, from identifying all proteins present in a given sample at a given time to to a global molecular analysis platform addressing functional aspects of biological systems. What are the various types of proteomics? Let us see what is known as protein expression proteomics. Expression proteomics is the quantitative study of protein expression between samples that differ by some variable. Protein expression of the entire proteome or of subproteomes between samples can be compared. Information from this approach can identify novel proteins in signal transduction or recognize disease-specific proteins. Structural proteomics is the field in which the proteomic studies in that the structure of protein complexes or the proteins present in a specific cellular organelle called as cell map or structural proteomics. Structural proteomics attempts to identify all the proteins within a protein complex or organelle, determine where they are located, and characterize all protein-protein interactions. Structural proteomics was the recent analysis of the nuclear pore complex. Isolation of specific subcellular organelles or protein complexes by purification can greatly simplify the proteomic analysis, and these are found to be the examples. This information will help together the overall architecture of cells and explain how expression of certain proteins gives a cell its unique characteristics. Coming to the concept of functional proteomics, it is a broad term for many specific directed proteomic approaches. In some cases, specific subproteomes are isolated by affinity chromatography, and this is used for further analysis. This includes isolation of protein complexes or the use of protein ligands to isolate specific types of proteins. This approach provides a selected group of proteins to be studied and characterized and can allow important information about protein signaling, disease mechanisms, or protein drug interactions. What is cancer proteomics? Genomes and proteomes of patients suffering from specific diseases are being studied to understand the genetic basis of diseases. The most prominent set of diseases being studied with proteomic approaches is cancer. Proteomic approaches are being used to improve screening and early detection of cancer, which is achieved by identifying proteins whose expression is affected by the disease process itself. An individual protein that indicates disease is called a biomarker, whereas a set of proteins with altered expression levels is called a protein signature. For a biomarker or protein signature to be useful as a candidate for early screening and detection of cancer, it must be secreted in body fluids, that is sweat, blood or urine, such that large scale screening can be performed in a non-invasive fashion. The current problem by using biomarkers for the early detection of cancer is a high rate of false negative results. A false negative is an incorrect test, results that should have been positive. In other words, many cases of cancer go undetected, which makes biomarkers unreliable. Some examples of protein biomarkers used in detection are CA-125 for ovarian cancer and PSA for prostate cancer. Protein signatures may be more reliable than protein, or you can say as biomarkers, to detect cancer cells. Coming to the applications of proteomics. The applications of proteomics includes biomarker discovery, clinical applications, disease profiling and diagnostics, study of protein interactions, dynamics of signaling pathways. So what are the nutritional applications of proteomics? The nutritional proteomics is an emerging field in which these technologies are applied to nutritional research. It holds promise to profile and characterize dietary and body proteins, digestion, their absorption, identify biomarkers of nutritional status with relevance to health and disease condition, and of course to understand the functions of nutrients and other dietary factors involved in growth, reproduction and health. So what are the technologies that are involved in proteomics? Numerous reviews of proteomic technologies and applications have been published. More recently, Nature Methods indicated or dedicated a separate section on mass spectrometry, that is MS in short we call as, in proteomics, that gives an excellent overview on topics such as large-scale data generation, analysis and validation, elucidation of cellular network of protein interactions, mass spectrometric, top-down analysis of intact proteins and clinical research perspectives. The proteomics workflow essentially consists of sample preparation and protein or peptide pre-separation, identification and quantification. The latter two compass the complex interface between data generation and processing or validation. Despite tremendous progress at all levels of this workflow, the term proteome remains in contrast to the genome, a theoretical entity, because proteomic studies have to update and it is never revealed as an entire proteome at all. So how proteomics is used in nutritional intervention? Proteomics in nutritional science has the potential to deliver biomarkers for nutritional intervention and individual disposition, assess nutritional status at the molecular level, and discover bioactive food peptides and proteins. Food components may not only alter gene and protein expression but also target post-translational modifications. Diet-induced protein modifications can ideally be assessed by proteomic techniques. So what are the challenges in proteomic techniques? First, the technology platforms will profit from further improvements including advanced protein or peptide separation techniques, better depletion and enrichment methods, and more sensitive and specific mass analysis techniques. Secondly, bioinformatic tools to assess data quality and to convert data into interpretable information are advancing rapidly. New systems will be able to support the reconstruction of pathways and regularity reconstruction and also the networks even in the presence of fragmentary data, thus helping to integrate incomplete omic data sets. Thirdly, intelligent focusing on proteome subsets at the level of cell organelles and protein subclasses or the mass spectral level, that is proteotypic peptides and multiple reaction monitoring, will provide deeper insights into molecular networks. Coming to the fourth aspect, addressing protein turnover at proteomic scale, that is interpreting protein abundance changes as a result of protein synthesis and degradation, will add value to nutritional intervention studies performed with stable isotope labeled amino acids, peptides and proteins. Furthermore, such data should enable an improved correlation between transcript, protein and metabolite data as an increased protein synthesis rate, which is more reliable in the biological context as just the change at the transcript level. So lastly, dietary changes in humans represent rather subtle interventions resulting in many small rather than a few big molecular changes, rendering data interpretation to be a challenging one. Therefore, improved definition of human cohorts undergoing dietary interventions through proper genotyping can be expected to deliver clear readouts from omics applications. The future development and success of proteomics in nutrition and health will depend on several factors. The proteomic technology platforms, independent of their application, will benefit from ever-improving protein or peptide separation techniques, better depletion and enrichment methods, and more sensitive and specific mass determination and sequencing techniques. The analytical strategy bears great potential for result improvement. Intelligent focusing on proteome subsets such as at cell organelle, protein subclass or mass spectral level, that is proteotypic peptides, and selected reaction monitoring will yield less complex proteomes but provide deeper insights into molecular networks. The third area of platform related improvements covers the field of bioinformatics, that is the tool to assess data quality and to convert data into interpretable information. Current gaps in omics data sets may be elucidated by reconstructing pathways and regulatory networks, even in the presence of fragmentary data. Bioinformatics processing of protein and transcript profiles of normal and transformed cell lines indicates functional impairment of transcriptional regulators in buccal carcinoma. If these network reconstructing and motive elucidating tools prove to be successful, they may also shed a different light on the terms reproducibility and comparability of omics studies. Rather than searching for the same transcripts, proteins or metabolites found regulated between related studies, one may focus on the common motives behind these, often at first glance the divergent data sets. Proteomics will largely benefit from cross-correlation with gene expression analysis and metabolite profiling. However, interrelated timing of gene and protein expression as well as metabolite generation remains to be understood. One possible solution is addressing protein turnover at proteomic scale rather than taking proteomic snapshots. It has recently become possible to interpret protein abundance changes as a combined result of protein synthesis and degradation. Protein turnover experiments performed with stable isotope labeled amino acids, peptides and proteins has the potential to deliver a different quality of nutritional biomarkers. So what is the central dogma of molecular biology? It is basically the flow of information from DNA to RNA and to finally protein, as you see in this figure. It is nothing but a large scale study of proteome is called proteomics. As we have already seen, proteome is a set of proteins produced in an organism system or biological context. The proteome is not constant. It differs from cell to cell and changes over time. Within a given proteome, the number of proteins can be as large as 2 million. Can you just imagine? This figure shows the areas where proteomics is involved. Hope you are able to understand. The experiments on proteomics collect data on three properties of proteins: location, abundance or turnover, and post-translational modification. This deals with how they are expressed and how they are modified in post-translational modification. For example, phosphorylation and movement of protein between subcellular components, involvement of protein in metabolic pathways and how proteins interact with one another. High throughput technologies are used for investigation of proteome. Mass spectrometry based on tandem MS is used. Gel-based techniques like differential gel electrophoresis is also used. So how do we classify? As we have seen earlier, we can, of course, in a summary we can say as structural proteomics, expression proteomics and interaction proteomics. Coming to the significance of proteomics. Though the term was coined in 1994, studying proteins involve insight on how protein affects cell processes. The significance is to analyze the varying proteomics of an organism at different times in order to highlight differences between them. So students, to conclude this lesson on genomics and proteomics, unraveling the magnitude of proteomics and metabolomics pattern may likely to provide insights into a tailored approach to diet and health. The use of new and innovative technologies might provide needed insights into molecular targets for specific bioactive components and their influence on individual phenotypes. More research is needed to highlight this phenomena of new concepts of genomics and proteomics. Hope to see you in the next class.